So the goal of today is to make some cheese, or at least go through the process of making cheese. Pretty much is actually pretty simple. You have milk and you find a way to separate its whey from curds. And curds is pretty much its protein and fats and a little bit of sugar. So we want to find some way to do that. So in this process, you can see all three happening. You have bacteria, you have enzymes or rennet, and then right here it's going to get acidic, and we'll talk about how that's going to happen. Once all three of those processes happen, you're going to get your curds and whey, and then you can separate it either by filters or, in this case, stirring and cooking, and then you drain out all the water. Then they just press it and then let it age, and that's how you get cheese. Some cheeses are fresh, so you don't have to age it. Other cheeses you age for a couple years. Pretty much here's what milk looks like. Most of it's water, about 90%, 87%, and the rest you have sugar, fats, and protein. So that's why it's such a good source of nutrients for a baby, because it has everything. It has water, has fats, has protein, and it has sugar. So it's everything you need to grow. For a cow, its protein is broken down into about 20% casein and about 5% whey. And you have 30% fat and about 40% sugars. For humans, it's kind of flipped with the protein. We have more whey than we have casein. Now, how is all this initially floating in the water? Well, for the fats, they actually don't. So if you just get, I'm going to sneeze, I apologize. If you get fresh milk from the cow, it's going to separate. You're going to get your fat layer or cream layer on top, and then your fat or the rest of it on the bottom. But we don't want that happening with our milk, so they break it apart and blend it till the fat's really small. So just remember, fats are made of phospholipid bilayers. You have your core in the middle in this case, but then you have this phospholipid layer on the outside, so it makes a little bubble or micelle. And this micelle is going to be, the outer part's going to be keeping everything in, so it's going to kind of float in water. And if you make it small enough, it'll never separate. Now, the same thing kind of happens with proteins. Instead of a fat phospholipid bilayer that's going to keep the hydrophobic stuff inside, you have something called casein micelle on the outside. And the really cool part of it, it has these little tails, these protein tails, that also push away from other casein proteins. So instead of them grouping together, they push them apart from each other and stay suspended in the water. Well, the whole purpose of cheese making then is to get rid of those little tails. So here's a picture that shows the two different processes. One is the acidic process, and the other one is through the enzyme process. The acidic process reacts with the tails, and then you're going to have a bunch of hydrogens, ions inside of it, and because of that, it's going to have these proteins attract each other. Once you have enough proteins attract each other, they're going to clump together, get heavy, and they're going to coagulate or gel out of the liquid. The same thing can happen with the enzyme. It's going to actually cut off all these little tails, and without the tails, they don't push each other off anymore. So they also get together and form into curds, and then you can separate it. So both processes work. So if we go back to the very first part, this is something present in things like cows, and they have it in their stomachs to help them digest the milk. So we can isolate it and use it to make our cheese. At the same time, you have many different ways to make acid. In our case, we just added vinegar to our milk, which helped it separate into its solids and liquids. But you could also have bacteria growing in there. So if you have bacteria growing in there, what's going to happen is you're going to break apart the lactose sugar into galactose and glucose. And in the process, you create something called lactic acid. And this lactic acid is going to be the part of it that's going to break it apart, or break apart the little tails so they could kind of come together, and then you're going to have curds. So what's going to happen is once they lose those tails, they're going to group together, and you're going to form these curds, and then you can separate it. So that's pretty much cheese. It's just pulling out the protein and fats away from the water. And the cool thing about that is once the water is gone, really there's not much things could grow into cheese, especially the cheese that has the bacteria already in there. They're going to be competing it out. Sometimes you put penicillin on it to give something. Blue cheese has other fungus or bacteria growing in there. So the different cheeses will have different things growing on, and sometimes it's just from the environment. So that's kind of it for cheese making. I'm also going to post up a video to how we do it in class, and then also videos from the internet. So thank you.